All right, welcome everybody. This is week 3 topics, diversification of eukaryotes for principles of biology students. My name is Hunter Lee, your overcaffeinated host. Well, let's get going. All right, diversification of eukaryotes. What are eukaryotes? Simply speaking, these are organisms with a nucleus in their cell. These may be single-celled, these may be multicellular. So broadly speaking, that's what we're talking about. We're going to get into some distinctions here, what that means, but what that means in relation to other sort of organismal categories. And the trends to be paying attention to here are what necessarily must have come first to sort of result in the great diversity of life that we now see on Earth. So let's get started. This activity provides an overview of the early evolution of eukaryotic single-celled organisms. The first eukaryotes appear in the fossil record around 2.1 billion years ago, billion with a B. Current evidence indicates that eukaryotic cells arose as a result of endosymbiosis, sometimes multiple endosymbioses, among ancestral prokaryotes. The early eukaryotic cells that resulted underwent great diversification to become the 7 major groups of eukaryotes, and you can find these in your textbook. This activity is designed to help students begin to understand the vast diversity of the protists, as well as some of the evidence and thought processes that were involved in the development of the endosymbiotic theory. First question. Protists have been called a catch-all group. What does this mean? All right, so protists are a paraphyletic group. Let me draw a little family tree here. Paraphyletic group means that if you have an ancestor, and then you have branches on that family tree, paraphyletic group doesn't necessarily have to include all of the descendants, just a fair grouping of them. So that would be a paraphyletic group, includes the ancestor and some of its descendants. So protists are a paraphyletic group including all eukaryotes except fungi, animals, or plants. Including all eukaryotes except fungi more properly, animals, or plant kingdoms. Protists are themselves super diverse. They range in size from microscopic to macroscopic. Some of the large brown algae can be like 60 meters long. Many protists have a characteristic overall form. They have organelles with distinctive features, or both. All eukaryotes alive today, including all living protists, have a nucleus and endomembrane system. Endo meaning inside, and membranes, of course, being membranes, so many membranes inside the cell itself. So they have a nucleus, an endomembrane system, mitochondria or genes that are normally found in mitochondria, and a cytoskeleton. So all have a nucleus, endomembrane system, mitochondria, or at least mitochondrial DNA, and a cytoskeleton. what is happening here. All right. There are more than 100,000 recognized species of protists. Even though the organisms in this group are so diverse, we can recognize some common themes in their evolution, including the evolution of a complex cell structure, novel genetic recombination strategies, and complex life cycles. In addition, this group as a whole has great ecological importance. Provide at least 2 examples of protists from Chapter 27 in Biological Science, 7th edition, that illustrate each theme. So I've got some examples picked out here that we can talk about. I encourage you to go check this part of your textbook out and find your own examples for your own edification. But we're just going to pick a few iconic ones here. So let's look at complex cell structure. Here we have Euglenidia. Euglenidia contains a light detector, an eye spot. Let's see if I can actually share this part of my screen here. Euglenidia. Let me see if I can just... You cannot see that. Here, I can figure this out. Can I figure this out? There we go. Entire screen. There we go. There. Now you can see it. So this is a Euglenidia. Contains we have our light detectors, our eye spot, contractile vacuoles, pyrenoids, and chloroplasts. So chloroplasts, of course, are important for photosynthesis. So there's one example. We've also got paramecium. Very iconic. Paramecium contains contractile vacuole, oral groove, and a cell mouth. You can kind of see that in the smaller diagram here. Micro and macronuclei, and cilia. Cilia are these little protrusions on the outside of the organism that help with motility and sometimes grabbing onto food items. So cell structures, we have light detector, eye spot, contractile vacuole, pyrenoids. I actually don't remember what the function of a pyrenoid is. I kind of want to look that up. Pyrenoid is a microcompartment within the chloroplasts of algae and hornworts. Its known function is to promote photosynthetic CO2 fixation. Cool. And we have paramecium. Oh, excuse me. So this is actually, the micro and macronuclei is actually belongs in our next section where we're talking about novel genetic recombination strategies. But paramecium also belongs up here. Also have a contractile vacuole. We have an oral groove, a cell mouth, and cilia, and the micro and macronuclei. So in our next section, then, under novel genetic recombination strategies, that micro and macronuclei function in sexual recombination and reproduction. So the macronuclei function in growth and maintenance and asexual reproduction. So micronuclei function in sexual recombination and reproduction, and macronuclei function in asexual reproduction and growth. Another example, laminaria. These display alternation of generations between the gametophyte, or the haploid organism, and the sporophyte, diploid, multicellular forms. Alternation of generations between gametophyte and sporophyte, multicellular organisms, or multicellular forms. Next up, we've got complex life cycles. And the example I've got here is Plasmodium. So Plasmodium has a two-host life history and causes malaria. So it reproduces asexually in its human host and sexually in its mosquito host. So Plasmodium causes malaria. It's an easy way to remember that. You'll find it everywhere. Reproduces sexually in human host, asexually in human host, sexually in mosquito hosts. Next up is cellular slime molds. These have a life cycle that includes single-celled feeding amoeba. Let's see. Yeah. So these have a life cycle that includes single-celled feeding amoeba, and in the sexual phase, haploid amoebas can fuse to form a zygote, which immediately undergoes meiosis, which is pretty neat. Amoeba can also aggregate to form colonies, and the aggregate can develop into a stalked, asexual, fruiting body that gives rise to spores, which can survive in unfavorable environments. So really, really interesting kind of diversity in survival and reproductive strategies here. So how do we want to write that? Life cycle includes single cell, let me make sure I spell this right, amoebas in the sexual phase. I just about had a heart attack and thought I might have forgotten to unmute my microphone. All right. In the sexual phase, haploid amoeba can fuse to form zygote can also aggregate to form fruiting body. I'm going to do this next one in a different color just so we can differentiate a little better. Why are all of my pen tools? There we go. All right. So ecological importance. So all the species of photosynthetic protists in association with cyanobacteria provide the basis for the food webs of all freshwater and marine environments. The absorptive or fungus-like protists aid in the decomposition and recycling of organic matter, organic matter being all carbon-based compounds in all ecosystems. So we've got all species of photosynthetic, that is deriving energy from the sun, from light, protists, provide basis of all freshwater and marine environments. Absorptive and fungus-like protists aid in decomposition of organic material. Cool. In the late 1960s and since, Lynn Margulis provided considerable evidence for the endosymbiotic theory of the origin of various organelles in eukaryotic cells. What is the endosymbiotic theory? So we can kind of break this down just by taking a look at the word here. We have endo, meaning inside, and symbiotic. So symbiotic organisms are organisms that are mutually beneficial for each other and living in association with each other. So we have symbiotic organisms, one inside of the other, simply put. So what is the endosymbiotic theory? This proposes that eukaryotes, remember these are organisms that have a nucleus in their cells. Eukaryotes originated when ancestral prokaryotes, most likely an archaeal species, engulfed formerly free-living prokaryotic cells and enclosed them in vacuoles. These endosymbionts became major eukaryotic organelles. So you had a prokaryote, that is an organism without a nucleus, absorbed another, and then that became a permanent feature in that combined organism's DNA that was then able to reproduce to future generations. So let's write that down. Endosymbiotic theory proposes that eukaryotes emerged from an ancestral prokaryote that engulfed formerly free-living prokaryotic cells and those became eukaryotic organelles. Which two eukaryotic organelles were proposed to have arisen from endosymbiont? Were proposed to have arisen as endosymbionts? And what evidence did Margulis present to support each organelle as an endosymbiont? First big one is mitochondria. Mitochondria, you might remember, is the powerhouse of the cell. has those complex infoldings, those cristae, for increased surface area. Mitochondria are bounded by two membranes. In addition, they contain their own circular DNA. So I'm going to go ahead and draw that in there as well, which is important because the rest of these cells do not have, the rest of the cell does not have circular DNA. So mitochondria are bounded by two membranes, and in addition, they contain their own circular DNA with no associated histones and ribosomes. Some of the inner membrane's proteins are more similar to those of prokaryotes than eukaryotes. So, double-bounded membrane. That's point number one. Two, they contain their own circular DNA and ribosomes. And point three, some inner membrane proteins are more similar to those of prokaryotes than eukaryotes. What is another organelle very similar to mitochondria that may have arisen from an endosymbiont? This one is more commonly found in plants. And that would be chloroplasts. So very similar to mitochondria, they have a lot of membrane surface area that feature these proteins that generate a lot of energy from their inputs. So, So we have our stacks of thylakoids, we have our inner and outer membranes, the stroma being the space in between the thylakoids. All right. Chloroplasts are also double membrane. So the double membrane is important because it suggests that a organism with a single membrane was absorbed by another, and that engulfing organism created a vacuole, or not a vacuole, a vacuole from its derived from its own membrane, which nested with the single membrane of that original engulfed species. And this is in part evidence of that engulfing, these two separate organisms that then became one. So double membrane. Chloroplasts also have their own circular DNA, very much like mitochondria, and ribosomes, and some of their inner membrane proteins are also similar to those, more similar to those of prokaryotes than to eukaryotes. It's important that it's the inner membrane proteins that are more similar to prokaryotes than eukaryotes. So if I have proteins studded on my inner membrane that don't exist on the outer membrane, that's the evidence. So if my light blue proteins are my prokaryotic ancestral proteins, and then my purple proteins are only on that outer membrane, and those are more associated with eukaryotic membrane proteins. That again is that emphasis of an ancestral separation between those two organisms. Question 4. The chloroplasts of dang it. Disycristata, chrome alveolates, and rhizaria are all thought to have arisen as a result of secondary endosymbiosis. What is secondary endosymbiosis? Again, we can kind of figure this out by just looking at the root words. So endosymbiosis, of course, was that endo, inside, symbiosis, mutually beneficial organisms. And then secondary would be after the primary, after the first the first endosymbiotic event. So secondary symbiosis is said to occur when a heterotrophic eukaryote engulfs a photosynthetic eukaryote. So both are eukaryotic organisms at this point. So this is when a, let's see. This is when a heterotrophic eukaryote, that is one that produces, that consumes other organisms to produce energy, engulfs a photosynthetic eukaryote. Photosynthetic meaning that it is autotrophic. It produces its own energy from sunlight mostly. Heterotrophic eukaryote engulfs a photosynthetic eukaryote, which becomes an endosymbiont. What evidence supports this idea of secondary endosymbiosis? Evidence includes the following. We've got chloroplasts that are surrounded by 4 membranes. Remember earlier we were talking about 2 membranes being evidence of that initial endosymbiosis? These chloroplasts would have 4 membranes. It carries out photosynthesis in its own plastids, which is super cool. So chloroplasts surrounded by 4, I just forgot an entire letter there, membranes. Question 5. Structurally, Giardia lamblia lacks complete mitochondria and was once thought to be an example of what the earliest living eukaryotes might have looked like. I'm going to pull one of these up for you because they're kind of iconic. And CDC has some cool just had one. Here we go. Nope. Ah, here we go. That's the one I want. Giardia. Structurally, Giardia lamblia lacks complete mitochondria and was once thought to be an example of what the earliest living eukaryotes might have looked like. If Giardia is similar in structure to the earliest living eukaryotes, what does this imply about the order of evolution of the various eukaryotic organelles, that is, nucleus, cytoskeleton, mitochondria, and chloroplasts? So Giardia has several flagella, these structures, these external structures for motility, for moving. And it also has a double nucleus. So you can see these double nuclei there. However, it contains no plastids and no functional mitochondria. So Giardia's structure indicates that the double nucleus in eukaryotes evolved prior to the endosymbiosis of mitochondria or chloroplasts. Let's write that down. Giardia have flagella and a double nucleus. However, it contains no plastids, and no functional mitochondria. Functional mitochondria. Giardia's structure indicates that the double nucleus in eukaryotes evolved prior to the endosymbiosis of mitochondria or chloroplasts. Prior to endosymbiosis of mitochondria or chloroplasts. We've since discovered that diplomonads, such as Giardia, and the parabasalids, the other group of excavates, have modified mitochondria. What types of mitochondria do they have? Excavates contain nuclear genomes containing genes that are normally found in mitochondria, or unusual organelles that appear to be vestigial mitochondria. Excavates contain nuclear genomes containing the genes found in mitochondria. Or unusual organelles that appear to be vestigial mitochondria. So vestigial simply means non-functional. So an ostrich has vestigial wings, meaning wings not capable of flight. Though they might have some other function, an emergent function other than flight. What does this evidence indicate about the evolution of the various eukaryotic organelles? So these observations, that excavates contain nuclear genomes containing genes that are normally found in mitochondria, support the hypothesis that the ancestors of excavates had mitochondria, but that these organelles were lost or reduced over time. Suggests ancestors had mitochondria, ancestors of excavates had mitochondria, but that these organelles were lost or reduced over time. That were lost or reduced over time. And we're talking evolutionary scales, periods of time, which for simpler organisms would be a lot faster. All right. Question 6. I love this one. The metabolic pathways of organisms living today evolved over a very long period of time, undoubtedly in a stepwise fashion because of their complexity. Considering everything you have learned to date about the evolution of life on Earth, put numbers 1 through 5 in the following blanks to indicate the order in which these organisms might have evolved, and explain your arrangement. And note that some steps happen at the same time and will be part of the same stage. I love this question for a lot of reasons. One, because it kind of forces you to be very methodical and sort of retrieving which steps necessarily had to come first, but also just from like a test-taking skills perspective, you can start to rule things out as being first or last pretty quickly. So for example, looking at my options here, I noticed that we have prokaryotes and eukaryotes listed. So looking at just at the first couple words of this list, I know that necessarily prokaryotic organisms had to come before eukaryotic organisms. And so my eukaryotes have to be at the end, and my prokaryotes are going to be the first half of this list. So right there I have my division. So let's take a closer look at these. We've got prokaryotes capable of performing the Krebs cycle, eukaryotes capable of performing the Krebs cycle, prokaryotes capable of performing electron transport, eukaryotes capable of performing electron transport, prokaryotes capable of performing glycolysis, prokaryotes capable of performing photosynthesis, and eukaryotes capable of performing photosynthesis. All right. Simplest metabolic pathway, most foundational, is glycolysis. Glycolysis in prokaryotes had to come first. And the way we know this, first of all, it's found in all eukaryotes and many prokaryotes. It takes place in the cytoplasm and can occur in the absence of oxygen. Because eukaryotes arose as endosymbionts within prokaryotes, the prokaryotes capable of photosynthesis and oxidative respiration must have evolved after the prokaryotes capable of those processes. So that's number one. Prokaryotes performing glycolysis. Requires no oxygen. All right, so next up, we've got a couple possible candidates. This again is that footnote here for some steps may have happened at the same time. We're still in prokaryote land. I'm going to highlight these in blue. Second, we would have prokaryotic photosynthesis. This requires an electron transport chain. Yeah. Prokaryotic photosynthesis requires electron transport and produces oxygen as a byproduct. Neither the Krebs cycle nor the electron transport can occur in the absence of oxygen, so necessarily that had to come next. Electron transport is required to convert NADH to NAD+, because glycolysis produces 2 net ATP and 2 NADH. The addition of electron transport represents an advantage. Organisms can then gain 8 ATP net from glycolysis plus electron transport. So we've got prokaryotes capable of performing photosynthesis, thereby producing oxygen, and prokaryotes capable of performing electron transport could have happened around the same time. They kind of facilitate each other, right? Finally, now this is where the test-taking strategy kind of comes in. We've got the first couple in our list. We only have one option left in the world of prokaryotes, and that would be prokaryotes capable of performing the Krebs cycle. So just process of elimination, we can kind of get there. The Krebs cycle in prokaryotes cannot occur without a mechanism to convert NADH to NAD+. Electron transport must have evolved before the Krebs cycle. And then finally, we're in eukaryotic land. So now we've got, this is post-endosymbiosis. So next up we would have eukaryotes capable of glycolysis, the Krebs cycle, and electron transport, such as oxidative phosphorylation. These are all thought to have evolved due to endosymbiosis of an amitochondrial organism and an aerobic bacterium. So amitochondrial, A meaning not having a mitochondria, a mitochondrial organism, and an aerobic consuming oxygen for energy, or requiring oxygen for energy, bacterium. So eukaryotes capable of performing the Krebs cycle, eukaryotes capable of electron transport. And then finally, of course, we have eukaryotes capable of performing photosynthesis would have necessarily happened last. Some of the eukaryotes with mitochondria are thought to have undergone another endosymbiosis with a cyanobacterium to produce chloroplasts in photosynthetic eukaryotic cells. All right. And that's our live stream today, folks. Make a good faith effort, fill out your worksheets, find some examples that are interesting to you for the above portions where we were talking about different eukaryotes to highlight these different themes of novel genetic recombination strategies, complex cell structure, life cycles, ecological importance. Find examples that are relevant to you. It'll help you remember it better. And again, good faith effort on the worksheet. Submit it for that extra credit. This will go live on the Learning Center's YouTube channel immediately, as soon as I have an opportunity to upload it.